for spinal cord injury, a very hot topic nowadays. Thank you.
Thank you very much. Sabah al khair. Good morning, everybody. I'm so happy to be here in Saudi Arabia, and I thank the Saudi Spine Society Scientific and Organizing Committee for this very gracious invitation. I'll be talking about brain-computer interfaces this morning, and just to give a broad outline of my talk, I'll initially be talking about the background of brain-computer interfaces and the work that's been done at other centers before going into my own research. We have a clinical trial now at the University of Ottawa that I'm primary investigator on. However, we're currently in the recruitment phase, so I'll be presenting my animal research with the non-human primate, the monkey model. So we all know why we're here. We've all seen patients like this that come in with a complete neurologic injury or a very severe incomplete neurologic injury, and the outcomes neurologically are not great. We know what to do the night of, but we don't always have the best solutions for years afterwards. Brain-computer interfaces have many applications. However, this morning we'll be talking about simple assistive control devices like a robotic arm for spinal cord injury. I will not talk about the vast literature on things like decoding speech for people with stroke. There are a variety of signals that can be used to drive a brain-computer interface. Now, I realize that this is a spine society and that there are people with a neurosurgical background and also are psychologically more stable brothers with an orthopedic surgery background. I'll try to keep this at a level that I think is applicable for everybody in the audience. There are signals that you can acquire from the surface of the brain, which is the EEG, but this is a noisy signal. And then right on the brain, there's a signal called the ECOG signal. This is often used in epilepsy, but even this is more noisy. So if we implant microelectrodes in the brain, we can get actual spikes and also local field potentials, LFPs, which are more higher signal-to-noise ratio and a better signal. These are the signals that we use surgically implanted electrodes for these types of research. This is a picture of the Utah array, which is the only FDA, well, actually that's not true. As of a few weeks ago, Neuralink, Elon Musk's company, is FDA approved, but they have not done any human trials with it yet. But this is the device that we implant. It's 4 millimeters square with 1 millimeter or 1.5 millimeters electrodes. And this is the one that I'll be using in the human study. And this is the one also that we used in the monkey studies that I'll be presenting. So the basic problem in brain-computer interfaces is that we get these multiple recordings of neurons, and we have to somehow convert this into a motor signal to drive a prosthetic device. This is a slide that shows the basic understanding. It shows graphically what we do mathematically, and I'd like to take a moment to explain this. If you imagine that there's only 3 neurons, you can then plot the activity of these 3 neurons in a 3-dimensional space. And then the next stage on the next row, we can project this onto a lower dimensionality manifold, like a single plane. This is called dimensionality reduction, and it's a key thing that we do. And what it allows is to take multiple noisy inputs and get it into one more statistically reliable input. And then we can smooth it, and this creates what we call a neural trajectory. It's like a mathematical representation of the brain activity, and it's more stable. So I'll be referring to dimensionality reduction and neural trajectories later in my talk. This is some research that was done over 10 years ago. On the top left, it's the BrainGate clinical trial, which I was involved in. And in the bottom right is the Pittsburgh trial. Even though these were done a year apart, I like to present them together because it makes kind of a brain-computer interface robotic race between the two candidates, both with severe spinal cord injury. So this was where things were at, very kind of slow robotic control that was achieved about a decade ago, and I'll show some of the advances that have been made since then. The first advance, which was really promoted by the Pittsburgh group, was to try to add sensory control. We all know that spinal cord function, control of our body, involves both motor controls, sensory controls, proprioceptive controls, which is a type of sensory input. And so in Pittsburgh, they had this idea that they would implant arrays both in the sensory cortex and the motor cortex, and they would stimulate in the sensory cortex as a result of sensors on the robotic hand. And what they showed, first of all, was that by injecting current in sensory cortex, they could change the activity in motor cortex, both increasing it and decreasing it. And furthermore, in this study, they were able to show that if they had a movement of a robotic hand, say the thumb or the index finger, this would map to an activity on the motor cortex that was the same region that was activated if they actually touched the participant's hand. The participant in this case had active sensation, and they were able to replicate the same region of activity on the brain. So they knew that they were actually able to do something very similar with the robotic proprioception than what the patient's own endogenous sensory control would allow. And what they showed here, if you look on the right, with control, they were able to decrease the amount of time that it takes to do a robotic activity. So I'll show a video of this. I'll let it run for a little bit. In this trial, they were able to show a number of different tasks with different type of objects. On the left side is with feedback, on the right side is without feedback in the same participant. And this is the fastest trial, but they also did the same thing for the mean trial length, where they were able to show significantly better ability to perform a task of the different sizes. This video goes on for a little bit. I want to wait for my favorite one, which I also felt so bad for the robot without sensory control, because it completely failed. Let me see if it's, I think it's around here. I'm not able to advance the video, but there's one where it tries to pour a glass of rocks into another glass, and the poor robotic interface without the sensory control was completely unable to perform the task. This is the one. You can see how lack of control. It's almost as if the robot is dysmetric without the sensory control. And there you go. So the next concept that I wanted to introduce is functional electrical system. A functional electrical system is a system whereby, even without brain control, you could develop a system that stimulates a muscle of somebody that has lost control of that limb, or a nerve directly, and create some kind of movement in the arm or hand. And so this type of system marries very well with a brain-computer interface. This was a trial that was published in Nature in 2016 out of Cleveland, where this patient has a spinal cord injury, had some control of the hand, but not enough to actually use it in any meaningful way. And the muscles are being stimulated and controlled by a brain-computer interface you can see in panel C that's plugged directly into this participant's brain. And so the brain signals are driving the functional electrical system in his own hand and allowing him, using his own brain, to perform all of these activities with his hand that he was otherwise unable to control. The last advance is a new advance and a new concept, which is a brain-spinal cord interface, and also published in Nature very recently in June. The investigators in this study implanted an ECOG electrode, so a surface electrode on the brain, and have a decoding computer, and then stimulate on the spinal cord using a typical spinal cord stimulator, an epidural spinal cord stimulator. And so they have these two cortical implants that are bilateral, and you could see a regular type of spinal cord stimulator that many of us have implanted. And so there's first a calibration phase. The calibration phase, they're correlating the brain activity with the types of outputs that they want to have. And so in this case, they're looking at iliopsoas and hip flexion, so they correlate it with the actual iliopsoas EMG activity, but also with the amount of torque that's generated in the hip flexion. And you can see after calibration, there's a very good decoding accuracy. And then they did this for a number of other muscles that are involved in locomotion. And I'll show the result as a video. This is a participant who has not been able to walk for 10 years and was able to develop some kind of locomotion after, I think, about 6 months or 7 months of rehabilitation following the implantation of this brain spinal cord interface. So very exciting developments, and there's only one participant, and this participant previously was involved in their previous arm of the study, which involved spinal cord stimulation only. So this is a field that's emerging and developing and obviously needs to be verified with a population. Our study is called Neurocognitive Communicator, NCC 1701, because we had the REB approved in January of 2017. And our goal is to create a cognitive neuroprosthetic. So instead of a prosthetic that maybe decodes the moment-to-moment activity in the motor areas of the brain, one that actually can decode the cognitive concepts. And so cognition in the brain is in the prefrontal cortex. And so we've been working for the past decade in the monkey prefrontal cortex to prepare for this human trial by trying to understand the physiology. I'll show some of these results. This is area 8AV, which is very small and easy to identify in the monkey. It's a little bit larger and more difficult to identify in humans. We have a approach that we're planning to do in the human population, but I'm showing the monkey data right now. So in the first experiment, we have, you can see where the array is implanted in the two monkeys. The task is very simple. The monkey fixates and then does one of 8 different movements. And we're going to try to decode which of these movements we could decode prior to the actual movement. So this is the ability to predict. And so we did a number, this is Chad Boulay is one of the postdoctoral fellows in my lab. We applied a number of different AI techniques, which are the different points in this graph. And you can see in many of them, we were able to achieve almost 100% decoding accuracy. We then repeated this study, but using the more robust local field potential signal, which we would be able to have years after implant. Spikes is a little bit finicky to obtain. So we were able to really demonstrate that with 400 milliseconds, we were able to achieve almost completely identical results. And this was conducted by my PhD student, Renee Johnston. We then asked the question, okay, so that's movement, but what about things like learning and learning new stimulus response associations or forgetting? What about this kind of rich cognitive signals? Can we employ these? And so this is Ali Reza Razidalab, is a PhD student in my lab. Actually, he just graduated his PhD. Looked at an experiment that we designed in which one of 2 cues, different colors, in this case green or red, are given, and only one of 2 different targets and distractors appears to the monkey. And based on the cue, the monkey has to decide which one the monkey will move to. And if the monkey gets the wrong one, it doesn't get its reward, which is some juice. And so every time the monkey learned it, we reset the cue association and frustrated the monkey. And so you can see this is the performance of the monkey. In red is where the monkey has not learned the task. On the y-axis is the performance. And green is where the monkey has learned the task. And then we give it a new set of cues and reset everything. So we want to see, can we capture this idea of learning? Are there neurons that can learn like this? And so we mapped the neural spaces and looked at these neural trajectories. And what we found was when the monkey doesn't know the cue, in the neural space, AI algorithms would not be able to distinguish between the neural activity between which response the monkey was going to make. However, as the monkey learned the association, the AI algorithm from the neural activity was able to dissociate these activities. And what this teaches us is that this area of the brain is involved in learning. And then we looked at the actual neural trajectories and mapped them up as points in a space, and we found that the monkey, the neural space was able to distinguish between every single different direction and every single cue. But did the monkey have this ability? Well, we looked at the monkey's performance on the bottom left and found that the monkey was able to remember different cues if they were applied later on, but would forget them if a number of different cues had been applied overhand. And when we looked at the neural space, it was the same pattern. And so the monkey remembers and forgets as the neural space AI algorithm was able to remember and forget. Well, this also demonstrates that this area of the brain is involved in learning and forgetting and being able to remap and be very plastic, which is something that we want in a brain-computer interface, because it may have different applications. If a person's using a robot, we go to one room or another room, or there's different context, we want it to perform in a different way. The last task that I'll show in this set of experiments was also done by Renee Johnston, where we put the monkey in a maze, virtual reality maze, and we were able to decode the location of the monkey in the maze with this activity. So this area of the brain is able to map location and able to map the types of activity that the monkey is able to do in learning, also predicting movement. This is what I've shown in this summary slide. So in our human study, we're planning to record both in motor cortex and also in this cognitive area of the brain, the prefrontal cortex. And we'll record from the neurons. It goes to a digital hub where the signals are digitized, a signal processor, and ultimately a computer where our decoding algorithms are written. And this is the equipment that we obtained through a Canadian Foundation and Innovation grant with the robotic arm, virtual reality, and all the neural signal processing equipment. And this is my last slide. This is Ali Reza Rosita Lab's next project is to try to, with the human participant that we'll be recruiting in the near future, we'll be looking at perhaps a two-stage algorithm where the cognitive array, the prefrontal cortex array, makes decisions. Like if you have, let's say, a coffee and a cookie, it will be able to decode what the person wants, coffee or cookie. And then the robotic arm will then operate from the motor cortex under a restricted space, which could make it even more efficient to actually perform this task. So these types of hybrid algorithms using these different rich brain areas will be what we'll be conducting in the human trials. And inshallah, next time I'm in Saudi Arabia, I'll be able to show these human results. I'd just like to acknowledge the efforts from the people that work in my lab and also my collaborators, Dr. Julio Martinez at Western University. Thank you very much.